Also, I'm so happy that Chandrima, Shinjini, Sandhya, and these people are here in this in the audience. And so, ladies and gentlemen, I'm not presenting the work which I have been doing throughout my career. Rather, I have decided, I was asked because Professor Jagannathan told me that it is better to speak some experiences during pandemic, whatever we have been able to do. So when I was at THSTI, I was approached by Ayush secretary because we had plenty of models to do preclinical evaluations for the existing formulations which are there and check, are they working for COVID-19 or are they working against SARS-CoV-2 induced pathologies. So during that time, we were nervous because these formulations are actually prepared in many different ways, and sometimes they are not properly characterized. So these are issues which we actually discussed, and they were addressed, and then following that we accepted to conduct these studies. So I am actually presenting on behalf of several of the scientists because they were co-PIs on the project, and it has been done by a large group who conducted and participated in these evaluations. So it's not moving. Yeah. So just to touch upon just to recapitulate again how this virus attacks and then what does it lead to. So we know that this enters through, so I think it is not working. Is it? Okay, sorry. So it enters through the nasal route, affects the lungs, and generates a lot of ROS. They lead to apoptosis and cell death of these cells. And then there is an inflammatory response. A lot of inflammatory cells migrate. It leads to immune response. And the cytokine storm, if it goes beyond a level, then it leads to multiple pathologies, affects major organs, and then eventually there is a lot of morbidity and even mortality was associated with this. So what we did, we actually used in vitro as well as in vivo models here. We had a hamster model established there, and we also had the transgenic mice model. We also used lung epithelial in vitro models to induce pathology and see that how their cytokine response or inflammatory responses are affected. We used again some animal models for this. Then we used in vitro cells, some primary cells as well as cell lines. So neutrophils, macrophages, and T cells were used, and we activated them by using either TLR ligands. So mostly we tried to use TLR 7-8 ligands, which lead to cytokine production, and then in presence of these extracts, how are they being affected. We also used some of these animal models where we wanted to see that whether per se these extracts have any pharmacological activity or not. So as you know, at that time there was these multiple options which were trying for the therapeutic option, like repurposing of the existing drugs. So a lot of drugs which were available and were tried there. Then scientists also, as the MPRO and other spike protein, these structures were available, so used in silico approach to screen compounds. Several FDA libraries and several other libraries were tried. Then in vitro screening for virus direct inhibition, therapeutic antibodies were actually used, and also a lot of natural herbal products were tried at that time. And the idea is to do in vitro model animal models, and then associated PK studies to conduct so that you get an idea whether there is any interaction, herbal drug interactions or not. So we also use this. We were provided aqueous extract of Withania, Dinospora, Glycyrrhiza, and Piperlongum, which was used in combinations. IU64, which was very well used during COVID times. And it has actually, in this formulation, there are 4 extracts, like Elstonia, Schwarzschier charaita, Picorrhiza, and Caesalpinia. Like these 4 plants extracts are making this formulation. And we were also provided 4 oil formulations, which were used through nasal route. So I will be actually concentrating mostly on Withania, because this is a very well studied plant, and it has been known to work against infectious as well as several non-infectious diseases. So today, like I am concentrating and presenting most of the work which we conducted or we performed with Withania extract. Although like parallel studies were conducted with most of the formulations which I have told you just now. So what we used here was the root powder, which was extracted, and it is an aqueous extract which was used. Because in Ayurvedic texts, aqueous extracts are mostly used for the treatment purposes. So this was prepared by a company in their GMP facility, and then it was given to us. Once it reached us, we also did our characterization by using LCMS. And then you can see that the presence of these compounds was there in these extracts. And we continue to look at the stability of this extract every 6 months, so that whatever we are using is actually stable and it has similar fingerprint. So in this way, we actually analyze all the extracts which have been used in this study.
The storage is on the same extraction or fresh extraction?
No. The same extract was used throughout the study, but in between we kept on analyzing its stability by using these studies and by using marker compounds. So first I will tell you about the antiviral efficacy of Withania, and here I am also giving a comparative data of Dinospora and as well as Dinospora Piperlongum combination. That is how it is used and prescribed to the patients. So we used a hamster model, and in this hamster model, like this is a prophylactic treatment, we gave this extract for 5 days and then challenged the animals with SARS-CoV-2 virus particles through intranasal route. And what happens that in most of these animals, we develop pathology, lung pathology, and we also check the viral load here, and also there is a decrease in the weight of the animals. So as you can see that in infected animals, there is significant reduction in the weight. And when you treat with different formulations, and here I will also like to mention that remdesivir we used as a control. So in all the experiments, one or two drugs were used as a positive control. And as you can see that remdesivir protected against the decrease in body weight. And even our Withania treated animals also showed that there was significant prevention in the body weight loss. So they were protective, while Dinospora and the combination were not as effective as Withania. And in the viral load also, you can see that in Withania treated animals, there was a reduction in the viral load as well. Although like in vitro experiments, when we conducted studies with the lung cell line infected viruses, we could not see so much of protection, although like 50% protection we could see. But as we understand, 50% protection is not a very significant amount of protection. Now, we know that in SARS-CoV-2 infected persons, there is a severe lung pathology. So in these animals, we also conducted the evaluations on the by histopathological parameters, and these were evaluated at ILBS, and they were blinded. So there was no bias. So as you see that in uninfected animals, there is a normal tissue, but in infected animals, we do see a lot of pneumonitis, then epithelial injury, and bronchitis also. So, and there is migration of several inflammatory cells. But in remdesivir treated animals, there was good protection, and same was also seen with the Withania pre-treated animals. So as you can see that at when we evaluate different scores, like in pneumonitis score, you see that protection by remdesivir and Withania. Then in epithelial injury, again, similar level of protection is almost seen. In bronchitis score, again, there is a very good protection. And when we evaluate like lung injury, then inflammatory injury score, then different types of, and then different, by using expression like of these several enzymes, we could also find that there was a difference in the Withania treated animals. So it was very convincing that Withania is exhibiting good protection. So we decided that we will take up this extract for further studies. So here in these animals, actually in the infected animals, you do see that their spleen size also gets enhanced. And when we use the spleen size as a marker of protection, we do see that in case of remdesivir and Withania, there was significant reduction in the spleen size. And that is also evident by looking at the different cytokines and their expression. And this is the data where we have actually done the mRNA isolation and have evaluated like TNF alpha, which is also known in the patients to be increased, IL-4, IL-17, then different other IL-13, IL-10, like whatever we could do. And in the hamsters, we found that Withania, in most of the Withania treated animals, there was protection, although it was not as good as of remdesivir, but it was significant in comparison to the infected animals. So this was again a very convincing data. So we decided to do another study by using these transgenic mice model. And I would like to tell you that we have done most of the studies prophylactic. The reason was that these Ayurvedic formulations were given to the hospital staff, in the police, and those people who were on duties. So it was being given to them in a prophylactic manner, and they wanted to know, like Ayush Ministry wanted to know whether these persons are getting protected or not. So most of these experiments have been conducted in that manner. So again in mice, we gave these extracts at the human equivalent doses. We used human equivalent doses because they were already in human use. So in these animals, if you see, again, in the lung pathology, gross pathology, it looks reduced damage. Again, in the body weight reduction, which is a parameter in all the animal models, there is significant reduction in the infected animals, and Withania could offer some protection. It was not as protective as it was in the hamster model. And you can see that the duration of, in this transgenic mice, actually all the infected animals die within 5 or 6 days. So there was an increase in the survival time. But animals still did not survive like it was surviving in the remdesivir group. So there was some level of protection even in this animal model. And you can see that even the viral load was slightly reduced, but it was not as good as remdesivir as we saw in the hamster model. Pardon?
Why it was so severe in mice as compared to hamster?
Because this is a very severe model. Hamster, actually in hamster we do not observe death. They actually revive after the infection. But in this animal, in transgenic mice, actually all the animals die. So it is a more severe model.
Part of the reason is artificially overexpressed.
Yes, yes.
It's
Yes, yes. Human receptor is, this is a transgenic, na? So ACE2 is expressed in the lung tissue and then more viral load can happen here, unlike hamsters. Yes.
Did hamster or that other model which you developed, do you use BSL-3 or BSL-4?
Yes, yes, all the experiments in BSL-3 facility. THSTI has this, and this was performed by Dr. Amit Awasthi's group. He has done lots of studies, preclinical studies in.
Question. There are many pro-inflammatory cytokines and genes. They are basically overexpressing, especially if you see the cytokine stop.
Yes, they are.
Whatever cytokine, let me finish my question. Whatever cytokines you name it, I have not seen the level of IL-6, which is the most potent cytokine those patients who infected with COVID.
So, should I answer now? So, actually what happens in hamster, we do not have cytokine kits available because there is a limitation. All the reagents are not available for hamsters. While they are available in mice, we have done some studies on the cells in these mice also, that I will be showing the data. So, in hamster we did only expression. In expression, you have to really standardize a methodology when the expression is increased and when it is coming to a decrease. But that sort of time we did not have for each and every experiment which we were conducting. So at the time when we sacrifice animals after 5 days of post-infection, we measured the expression of those cytokines. So sometimes cytokines like TNF-alpha is showing beautiful induction. But in some cytokines, we did not see as good induction as we were expecting. Maybe it is coming earlier or maybe it is coming later. So we have not done such extensive studies here. I hope I have answered your question.
The extract which you are using, these are not in a single active molecular level. These are combination of
Yes.
multiple things.
Yes. So we wanted to use the extracts as they were being used or consumed by the human beings. So this Withania extract, aqueous extract, was being used also at that time during pandemic by human. And we used the dose which is human equivalent dose.
Another just quick question. Have you checked that heavy metal toxicity
Everything.
in the extract like lead, mercury?
It is extract which is prepared in GMP facility. So all toxin and heavy metals, everything is taken care of. Like there was no time, so I have not gone into those details. So again, like all the parameters which I was telling about hamster were also performed here, and we do see some protection even in the Withania treated animals. And so in some parameters, like in the inflammatory score, you see equivalent to remdesivir, efficacy equivalent to remdesivir. So this is the cell-based system which we have done. We also, like because of the paucity of time, I have not included all the data. But we have done very extensive studies here, and we are going to submit this publication very soon. So here, like if you see that, like human subjects, there is variability in the T lymphocytes, but not in neutrophils. We do not see so much of reduction in neutrophils here. And also like in macrophages, NKT cells, there is not so much of change which we see in the human or in the patients, we did not see here. But we do see MDSCs and monocytes which are getting altered. And if you observe here, like in case of monocytes, this remdesivir and partly even Withania is working. So I will go into the in vitro data where we have conducted and simulated change by using these TLR ligands, that how are they getting protected against the induction in the cytokines. So if we summarize, I will be telling you this data subsequently, but in the data which we have collected for the cells, it seems that it is affecting all these responses, and the animals were protected against the viral particle, like there was decrease in the viral load, as well as inflammatory changes were also reduced. So I would like to tell you that during the time, there were parallel studies which were being done in the patients by Ayush Ministry, and they did compare the Vithania with hydroxyquinoline. At that time, this was like during the first wave, this was very extensively being used. And we could, like if you see, the conclusion was that we report that this Vithania was not found inferior to hydroxyquinoline, and the efficacy was within the 15% of non-inferiority. So the way they have compared, it was like comparative to hydroxyquinoline. Now we know that hydroxyquinoline was not acting at all against that, but at least during that time, it was no toxicity or no adverse effects were seen in Vithania group. So then there were parallelly studies were going on in other labs where they actually did simulation studies by using bioinformatics approach, like molecular dynamic simulation by using this software. They found that there is the Vithania, the compounds which are present in Vithania somnifera were showing some affinity for spike protein as well as for MPRO, which is actually a protease used for the multiplication of these virus particles. The next study, which is published in the Natural Products very recently, what they have done, that they have done again the similar studies, and they also conducted in vitro studies by using these proteases. And they could show that there is IC50 of 540 nanomolar and 1.84 micromolar by using these compounds. And in vitro, they also showed that there was some antiviral efficacy. Although while we were using, I told you that we were seeing around 50% protection, which is at the time was not considered at all active.
So this you said that is, can you go back to this? You said this is extract, this is not a single molecule.
No, this is, I'm presenting some data from some other lab, not from.
Single molecule?
Yeah, this is single molecule. This is Vithaferin A, which is present in Vithania, and Vithanone. Like there are other reports as well that they are showing that this is actually in vitro inhibiting the enzymes which are responsible for the entry as well as the replication of these virus particles.
They are acting like a protease inhibitor which is associated.
Yeah. So as I have told you, like covered about the antiviral efficacy, and now I will switch over to the inflammatory response. So please tell me when I have to finish. So here, like when you see the neutrophil responses, we do see that it is showing some inhibition in the ROS generation, which is actually used inomycin PMA, that is a forbolmestrate, and also the TLR7/8 ligands. So when we, this is a TRLM, which is a ligand for TLR7/8 receptors. So these are the receptors which are used by the virus to actually infect neutrophils. So here again, we do see that these are the positive controls which are showing very good effects, but Vithania is not showing so much of effect. So in neutrophil responses, we did not find so much of inhibition of the free radical generation. There was partial protection and also against netosis. In case of this SARS-CoV-2 infection, in the pathology, in COVID pathology, neutrophil role has been actually proposed initially, and it is now proven that it induces the thrombotic response in the patients. So netosis, whether it is getting or inhibited, we also evaluated, but unfortunately, Vithania is not so effective against that. We wanted to check whether it is interfering with the antibacterial activity of neutrophils or not. So as you can see that Vithania did not interfere in the active antibacterial efficacy of the neutrophils. Now we wanted to see that, as I told you, the TH1, TH2, and TH17 responses which we see in the MIs we also wanted to see whether these differentiation were getting affected or not. So quickly I will like to add that here we used dexamethasone as a positive control, and if you see the Th2 differentiation IC50 was around this. So though it was effective, but not so much. And I am not very sure because we have not measured the Withania in the animal blood, so we don't know whether it achieves that level or not. Then we also perform such studies on the THP, differentiated THP cells, and as you can see here, that there is protection against TNF alpha, MCP1, IL-6 you were mentioning. So here you can see, Dr. Kundu, that there is a, yes. Yeah. So it is differentiated by BMA, and then we treat with Withania extract, and then induce. We have checked the thrombosis. We have checked thrombosis, but not the. I will show some data. So this is how, like in vitro, by using TLR7/8 ligand, we could see that there was protection against several cytokines in presence of Withania extract in vitro. Then I will come back to the lung epithelial cells. And here, like what we did, we used these cells, and we also induced them with this ligand. But unfortunately, lung epithelial cells did not get stimulated by TLR7/8 ligand. They did not induce so much of cytokines. So we used LPS here just to see a model whether it is showing any efficacy or not. And then we also used LPS-induced air inflammation in BALB/c mice. So quickly, I will like to tell you again, we measured several cytokines in this, and you can see that there was a very good protection by Withania. So we did not see so much in neutrophils or in the macrophages. We do see in Th2 cells, but in lung epithelial cells, it was showing very good protection. Sorry, I think I do not have so much of time, so I'm not going into more details. This is by the measurement of these cytokines by ELISA. So expression as well as ELISA results were similar. Because here we could do that at different time intervals. We could do some standardization and identify the optimum time. Now, this is again in THP cells because we were doing LPS, so we wanted to see whether Withania is working against LPS-induced cytokine induction. As you see, the data is very convincing that it is actually reducing LPS-induced inflammatory response. Now, in the animal model, we again used LPS, and you see that there is a reduction in the neutrophil count as well as in the total leukocyte count in the Withania-treated animals in a very dose-dependent manner. So 100 milligram was very effective. And these are all pre-treated animals. We have used only prophylactic way of looking at that. In some for half an hour before, or in some in 4 hours before, depending on the protocol which we are using.
The idea is to prevent, preventive.
Yes. It was being used as a preventive only. So this is again in the mice lung tissue, we again measured the cytokines. It was showing very good effects. And this is a chronic way of looking at, like in LPS, after 4 or 5 days, what was the inflammatory score? And it is a very convincing data that there is a reduction in the inflammatory score as well. So next comes some studies in the animal models. Like you can see that Dr. Kundu, you were asking some questions here. So I would like to say that in endothelial cells, in the aortic tissue as well as in the endothelial cell culture, we do see that there was good protection by Withania, and it was actually inducing a vasorelaxant effect. So it was cardioprotective, and 50% of these animals were protected against collagen and epinephrine-induced thrombosis. So again, very convincing that it has a cardioprotective as well as a neuroprotective response. So this is a summary. I have not gone into this data, but we already have, and we have already published this, that it does not interfere with these major CYPs, which are actually being utilized by the human liver tissue to metabolize most of the drugs. So there is not, I am not anticipating there is a potential herbal drug interaction if somebody is using ashwagandha or Vithania. So in general, like this is a literature review which I am showing you, that so many studies have been conducted and are reported in the literature that it actually interferes with the viral response, like entry and replication. There are in vitro indications, and our in vivo data also supports partially that yes, there is a reduction in viral load. There is immune imbalance which is getting corrected. Then cytokine storm is also getting neutralized. Hyperinflammatory response which happens in these patients is reduced. Then vascular permeability or vascular protection is there. Then inflammation and pyrexia. We did not do pyrexia, but here inflammation response was reduced. And in multiple organ failure models, people have performed those studies, there is also protection. So Vithania seems to act at most of the stages, and if these extracts are prepared and properly, and they are actually safe because they are in human use for a very long time, it seems that Vithania prevented a lot of morbidity which was happening in the patients. So I will like to acknowledge there was as I told you, it was these studies were performed in several groups, and I am presenting on behalf of them. So these were the scientists who have participated. Then there is one participation even from the Pune University. Some studies were conducted at CDRI Lucknow. I like to acknowledge the fellows who actually joined and left, like this is what is happening. So there were a lot of fellows, and these 4 are still there, and the people who offer technical assistance. So in addition, like directors from THSTI were very supportive to this project. Initially, Dr. Gagantipkang, and presently Dr. Garg. Then Ayush secretaries were also very supportive. They were very keen on knowing the data, so I was asked several times to actually present this. So thank you so much. No, I will not suggest to take it for years and years, but rather during COVID time, I think people took it up to 6 months.
Up to 6 months?
Yes.
So is that the longest duration that has
no,
been tested?
Actually, one has to see the published data. So when we see, like we want to see or we review the publications, we do not see such claims. But I know, like Ayush Ministry had discussed this, and they were saying that they consider it ashwagandha as the safest yes, formulation.
And since many of your effects are, I mean, assuming that the bioinformatics says something, many of your effects are very pleiotropic, I mean, across the whole innate immune response. And so would it not work against influenza or any other viral respiratory pathogen?
Actually, when we say the immune response, I will say that it will work against most of the viral infections, because it does not seem to be very specific for SARS-CoV-2. Although, like computer-aided drug design experiments are suggesting towards that, and there is some data which people have done by using MPRO and also the spike proteins, that there is some interference. So, but we really don't know whether the compounds which are showing to be, are they achieving that much of concentration in the human sample or tissue or not. So this is yet to be demonstrated. So I will say, I will agree with you, but there is indication from the human studies as well as from the animal models that there is a reduction in viral load, whether it is due to immune response or due to direct antiviral activity.
Dr. Madhu, one more question.
Yes.
Very interesting study. So this is from ashwagandha, right?
Yes.
If you look at, there are two Indian couple, they are in Japan last 30-40 years, and their whole life working on this. They gone all the way up to single molecule. And their major studies, they are showing in cancer model, it is showing anti-cancer activity. I think
Yeah, there are a lot of studies for anti-cancer. Yes.
So my suggestion, if some of that molecules already available commercially or some other researchers have identified, why not to use at the molecular level, maybe combination of 2 molecules or 3 molecules, and based on structure guided drug design, some maybe docking approach or other way, if you exactly pinpoint what is the target within the SARS-CoV-2, and then move it little further, maybe you can use some of the proteomics approach or some other approach as well.
So I will surely discuss and share your comment with Amit, because antiviral thing Amit has done. But for inflammatory cells, we have actually have been awarded one more project where we intend to do the compound related studies. So whether at the compound level we are showing or we are getting these efficacy or not. Thank you so much for. We have not done, but there are reports.
Others might have done.
Yes, yes. Britannia is relatively safe. Yeah. Provided they are like, these extracts are prepared in GMP facilities and metal contamination as well as bacterial contaminations are taken care of. So it is important that from where we are actually procuring this.
So Udania is available in different forms by commercial sources, even formulation or supplementation. So people are actually taking it.
People are taking it, but I will say that you have to be very careful from which
it's very in-depth scientific study.
Which pharma is preparing and now using. Right. So we have, like this has been prepared by Pharmenza, who have a GMP US certification as well.
Just as to respond to Sandhya's question, this is an observational study that has seen these effects. I think if one has to look at influenza or any other specific disease, it would be good to actually look at it in a more, in a randomized control trial where you actually see a cause-effect relationship. And that would be interesting because then you can actually evaluate the immunity or the host responses within the comparison group. So.
During COVID time, there was like
no. I'm talking about what
I got it.
Yeah.
But we were interested in doing some human trial studies as well, but it was impossible. So THSTI was overloaded. In fact, even conducting these studies took a lot of time, and there was a lot of turnover even in terms of fellows as well. Yeah, Ranga. Yeah.
Excellent presentation.
Thank you
My
so much.
only concern here is you are showing upregulation of several pro-inflammatory cytokines continuously, which is unusual for for the homeostatic reactions of any immune response. And that to people when they are taking continuously, you might argue that they are taking their safe. But how do you justify that level of high cytokine expression and that being good?
Yeah, this is not only your concern, we were also concerned, but as I told you, we have done most of these studies on single time point. We have not done a multiple time point study, which I would like to do. But there was no time to do this because all the studies were being done at BSL-3. And our facility at that time was smaller and very loaded. Lot of vaccine candidates and so many things were coming in. So if we do a time duration and then check whether every cytokine is getting reduced or not. So this is the reason we did in vitro studies where we did some optimizations in terms of proper induction at what time and then whether it is reduced or not. So if you see our in vitro data, there is a reduction. So I will say that let us not judge only by one time point study. We need more studies here to show efficacy. So thank you so much, and thank you so much for your interest in this.
Yeah, thank you once again, Dr. Madhu. So
Now, I have the pleasure of introducing the next speaker, Professor Shinjini Bhatnagar. We both were colleagues at Inst, Delhi. And then she moved as a dean to translation research, Faridabad. Now, after superannuation, still professor of eminence. She is going to talk on the multidisciplinary approach to diseases that affect the public health. Dr. Shinjini.
Hello, can you hear me? Yeah. How does this function?
It is on left and right.
This is further?
Yeah.
And the pointer is here.
The pointer button is
Okay.
laser.
Thank you. Thank you very much for giving us this opportunity to present this very unique, interesting, and yet challenging program, which is multidisciplinary approach, which is essential to understand complex disorders of public health significance. And I'm not, again, like Madhu, I'm representing a very large group, and I may not be able to answer some of the questions which are not really my domain expertise, but I'll try my best to do that. So, you know, when you think of science and technology, and we all know that significant discovery and transformative, impactful innovations is what actually is the key to interpretation of the complexity of our lives, our diseases. But as a physician scientist, I'm a pediatrician by training, and I see little babies. And I'm one of a small cadre of physician scientists in the country. And so for us, or for many of us, research which goes into clinical practice and policy becomes very important, because you can actually then apply that research to your patients and to the larger population. So when you're thinking of an idea, it's the discovery as well as the innovation. For us, the application that one is making is inspired by the need of a society. And that then becomes the application which can go back to the society for an impactful intervention. And because of this whole thinking and the philosophy, and again, being a pediatrician and I'm concerned about the maternal child health, which is the key determinant of the health of a society. Despite all the progress that we are making, our maternal mortality is high, and our infant and neonatal mortality is unfortunately still continues to be high. And as you can see here, that one of the important, there are many causes, of course, and I'm not going into details of that for brevity of time. Preterm birth, which earlier was believed to be a problem of the West, actually is a concern that we have. And some model data has shown that it's a large number of babies who are dying, who are born annually, die soon after birth, contributing 35% of total neonatal deaths and almost a quarter of the global deaths today, even today. And this is a huge concern. And not only do they die or have problems at birth, they continue in life with long-term disability, contributing to the morbidity in the society. And one of the sustainable goals that we have is which the government has set up, is to have this as low as 12 per 1,000 live births. And today, as you can see, it's much, much higher. So there are many problems. I mean, if I was to tell you, small for gestational age is 30% of the babies who are being born. And these are the ones who continue to either have undernutrition or go the other way and have metabolic problems that we are now facing. And we have almost 25% of babies being born low birth weight. So these are many problems, but we decided to concentrate on preterm birth. The reasons being that it's novel. It's believed that it doesn't exist in our country, in low middle income countries like ours, but it does. And the problem is that not much was known about it. And that came from so many unresolved questions and key challenges, because it's a very complex syndrome. It's not a disease, it's not a phenotype. It's a complex syndrome which can have many genetic, biological, psychosocial, and environmental factors contributing to the etiology. There are large gaps in understanding this disease. Whatever data that's available is compromised in sample size, is one time. Like Madhu said, it's one-time data which you cannot attribute a lot to. It's not as if you have the longevity of a patient being followed up. There's a lot of phenotypic heterogeneity. As I said, snapshot evaluations, not much available mechanistic pathways. And for this reason, we decided to take up this challenge and actually understand this disease in a time series manner, in a multi-dimensional approach. And we based it on, because we felt that the right questions were not being asked, maybe the right people were not asking these questions, maybe there was work in isolation, and again, were there gaps between evidence to clinical practice to policy, and therefore we didn't have this. And therefore, as I said, a combination of these sciences would bridge this gap and help us to understand the disease better. And we started this program, which is called Garbhini, the Interdisciplinary Group for Advanced Research on Birth Outcomes. It's a DBT India initiative, and here we had the fortune of having a secretary, Department of Biotechnology, who understood the challenges of this kind and was very supportive in helping us to start this, Professor Bhan. And this has now gone on to become the Atal Jai Anusandhan program of DBT and a flagship program of THSTI, because we are largely coordinating it. Having said that, THSTI plays some role. We have large roles coming in from NIBMG, Regional Center for Biotechnology, IIT Chennai, IIT Delhi, and above all, District Hospital in Gurgaon and other tertiary hospitals. So what is our mandate? It's bridging clinical epidemiology, multi-omics, data science to facilitate a translational pipeline. And what do we need? We need tools for early identification and prevention of a high-risk pregnancy that facilitates timely and improved care. And the reason why I say this is that when we get these babies, they're already born. So what you're trying to do is you're trying to protect a baby who's already diseased, and the cost of that, the trying to save such babies, is not easy. So we want to have tools where you can pick up these mothers early, do a risk stratification, and this risk stratification should come from novel predictors, using some of the old predictors and risk factors, but should come from novel predictors which are based on epi and clinical risk factors, ultrasound-based tools, and use multi-omics analysis for developing biomarkers which can thus go into this risk factor identification and into an algorithm for developing this tool, and use this opportunity for developing some mechanistic pathways for understanding what I said were the large fundamental gaps in understanding the biology of this complex syndrome. So what did we do? As I said, we wanted to study it in a time series event, a complex heterogeneous phenotype. We set up in a district hospital in Gurugram a clinical research unit, and the reason why we chose a district hospital and not the population, some of the true the hardcore epidemiologists turn around and tell us you should have used the population. Sure, that may be the next step, but at the moment we had to set up a system which could cater to a multi-dimensional approach. We needed some lab facilities, we needed an ultrasound facility to be able to bring out the initial key findings, which can then further move maybe into the population. So as you can see, we set up this center in the district hospital in Gurugram. Not easy, because there were no lab facilities and people did not believe that research can happen there. And yes, yes, huge.
And this district hospital, ESI or
No, it's not ESI. This is the district hospital which caters to almost 22 primary health centers across. So it caters to a rural and a semi-urban population which comes to this district. It's a secondary level hospital. It's not a tertiary level hospital. We did not go to primary health centers because we needed, when I talk about omics, we needed a lab facility which could do the initial work there. And that would not have been possible either in the population, because giving blood samples in the population of the kind we wanted, we are collecting 20 to 25 ml of sample from pregnant women across pregnancy for various parameters. So this would not have been easy in a population. Now, having had a setup in a district hospital, they know us, we can go into the population. And so we're doing other studies in the population. The disadvantage of a hospital is the attrition is very high. In a population, you may not have that high an attrition. They don't want to come back to a district hospital for a delivery also. So this was set up, and as I said, these are our partners, and these women are enrolled in the cohort early in pregnancy, definitely less than 20 weeks period of gestation, based on a dating ultrasound. So our ministry, DBT, supported us. We set up an ultrasound facility there, which is very high end, which could actually, as I tell you, has led to some most interesting image-based applications today. So they are followed up during pregnancy at regular follow-ups as part of their antenatal visit. One visit in the postpartum period. We collect multiple biospecimens from the mother, as in sera, saliva, urine, high vaginal swabs, and we are taking neonatal heel pricks, cord blood, placental tissue. And the more interesting program, which is probably one of the largest in the world today, is that we have serial ultrasounds of the fetus as the fetus grows across pregnancy, taking all uterine, placental, and fetal characteristics as we move on. So this is where we are today. We started in late 2015. Took us some time to set up the cohort. We had a COVID break. Our sample size, a priori calculation, was based on the risk factors that we wanted to analyze, was 8,000 mothers in the first phase, which is till December 2020, and our second phase is going on to enroll up to 12,000 women. Today we have 11,000 women enrolled in this cohort, and we have follow-up samples, and they're very well characterized, phenotyped biospecimens of almost 10,000 participants, several aliquots collected in various forms, as you can see here, of the blood, and of course ultrasound images. Now, this is all very well, you create a platform. This platform is it's one of the largest in South Asia, probably global. What do you do with it? I mean, this platform, and I'll come at the end of this, that we do, this is going to be, it is a national platform. We are now reaching out to other partners that we're very happy to collaborate for more questions. But where are we today? We've been able to give to the government, in a first time in the country, a systematic longitudinal analysis of preterm birth rate, which is my apologies, which is as high as 13.4%. It's huge. I mean, it's much more than what we've even expected. The hospitals are recording 5 to 6% because the documentation is not the best, which is understandable given that most of them are bursting at their seams. I'm talking about the government public hospitals. We saw 13.4% with, You can see how significant that is. And large number of them are spontaneous preterm births. And why I'd like to draw your attention, we don't know what the cause is. The mother comes, ruptures the membranes and delivers. We don't know what's happening there. And this is the problem that we need to look at. Look at the stillbirth rate, 2.6. You convert it in a country like ours with numbers, huge numbers. And you can imagine the economic situation as well there. You know, the mother has gone through that pregnancy, the antenatal, and most of them are antepartum stillbirths, small for gestational age. What we found is very novel. Antenatal vaginal bleed is something that has been seen, but biomass fuel use, passive smoking, obesity are actually risk factors for adverse pregnancy outcomes. Obesity in a country which is still malnourished. So that was very interesting. And we've been able to give this to the government. I'll just run through some of the clinical epi results because I think it's important for us to understand why this is. For the first time, we've created Indian population-specific gestational weight gain reference charts. And as you can see, our women, by the third trimester, 43% of them have poor weight gain. Half of our women don't gain weight well by the third trimester. I mean, it begins almost the same, and then the faltering happens, as you can see here. And this has great association with metabolic outcomes, which we are now trying to see during pregnancy and childhood. And we're incorporating community cohort data, as I said, to improve the representationness of this chart. But what does it mean? What does this gestational weight gain mean in terms of the adverse outcome? One, of course, is the maternal health. The other thing is, as you can see here, this U-shaped curve shows that those who don't gain weight and those who gain weight abnormally, both are at risk of producing preterm birth. And the interesting thing is that with these charts, and I'll just come to these numbers, you can see the gestational, actually the Z-score at which you can intervene with the mothers. So this is a straightaway application that can be done in the population. And this data is come not only from our cohort, but because we've harmonized with the other international cohorts. We, of course, are the largest by means of the population and the largest numbers. We've been able to, we have this data on 13,000 mothers. How many are malnourished? Well, 70% of our mothers are malnourished. I mean, 50% don't gain weight even during pregnancy. So I don't know where the improvement has happened. Somewhere, yes, but is it incremental? No. The other thing which everyone worries about is the air pollution, and there we've collaborated with IIT Delhi and IIT Madras, who are equal partners in this program, where we've seen the exposure to ambient air pollution of more than 110 microgram per ml in the first trimester reduces pregnancy duration. So the gestation goes down with an exposure to ambient air pollution, and a more specific result we've seen is that for this increase in the PM2.5 concentration, the cervical length reduces. And cervical length reduction is a pathway to an early gestation in pregnancy. And now we're trying to, with IIT Madras, we are developing sensors for personalized exposure in pregnant women to actually see what is happening at the household level. And this, and then translate it into a policy where you can use, go on to do intervention studies in the next phase. So the other thing which we've looked at is micronutrient, not going into greater detail, but two very novel micronutrients for anyone who's interested in nutrition here is copper and selenium. Selenium helps in implantation of the placenta, and we are seeing that we're doing that across pregnancy, but what we've seen with copper, in maternal copper, that an increase in maternal copper actually brings down the gestation. So that was something which was interesting. What we found is that gestational diabetes, everybody thinks GDM is routine, but is not so common in pregnancy, but not so common, but in a secondary level care hospital, we've seen it as high as 14%. Again, that's a huge finding, and we are now working more to look at, this is very preliminary just now, so I won't go into detail. So this was a kind of bird's eye view of some of the important clinically important risk factors that we found. We then put it, with our data science partners, put it into a prediction model. And first we used the conventional analysis, and we found that only 8% of our risk factors could explain the cause of preterm birth. And then we did a more advanced dynamic personalized risk prediction model, and when we went through the 3 trimesters, even as high as in the last trimester, it's only been, the ROC is, the area under curve is 0.75. So we are way away from actually picking up what are the predictors, and what does it mean? It means that you need to add more biology, you need to add more omics, you need to add more ultrasound images, you need to add more other multidimensional factors to be able to get there. And one of the factors that's very important is the actual phenotyping and dating of the baby. And there are 2 ways of doing it. One is, 3 ways. One is either you use the last menstrual period, very clinical, with a lot of subject expertise. The second thing is to use data from the ultrasound as the anthropometry, and third is the ultrasound images. And what we've done, again with our data science partners of IIT Madras, again that just shows how multidimensional one can get. We found that with our fetal, so again just to tell you that all the data of the way our babies are being characterized is Western data. All our ultrasounds have Western-based data for picking up baby sizes and baby gestation. We don't have our data. So what we've done is, with our own anthropometry of the 10,000 women, of the 8,000 women that we have, we've developed dating models, which now, as you can see here, these are our formula, and this is the Western formula. And these are the formula which are closest to the gold standard of the first trimester dating. First trimester dating is very close to the Western data because it uses the crown, it uses the head. But as the baby grows, we are smaller, and therefore we cannot translate our size as the Western size. And it's for decades that we've used this. I mean, I as a pediatrician have accepted it. So now we have this data which is very close to the gold standard, and this is the Western data, and this is the intergrowth, which is a more recent international. So we are much better, and I'll just come to this again. And the other thing is, we've actually used our ultrasound images of the head, femur, and abdomen, put them in the model, and use them to pick up the gestational age. Again, with our data science partners, and we've seen that the internal validation and the external validation both work very well. As you can see, this is the Western data, this is our data, which my colleagues have called GORGE, and this is, so the interesting part of this is that for people, I don't know if there are any data science people here, but what we've been able to pick up, that this model is picking up the babies who can have fetal abnormalities much better than any of the Western formulas. So this is, but having said that, we can't be complacent. We need to move forward. We need to validate this. And what we are now doing is validating it in external cohorts. We are validating it in CMC Vellore. We are validating it in a clinic in Delhi. And we are also going to Northeast, where we'll be validating these models and to see the representativeness of the model. And only then will we, and then something that we discussed in the morning, we'll go back to my colleagues, the radiologists and the gynecologists, and say, this is it. Are you going to allow this software to go into your ultrasound machines to be able to pick up your babies, because at the end of the day, they are the stakeholders and they are the ones. So we'll have to go through that exercise, and believe me, it's not going to be easy because there are regulatory approvals and then only the clinical deployment will happen. So another interesting thing is we're using video swipes.
What is the ultimate endpoint of that gauge?
So the ultimate endpoint will be that we will have, so what happens just now is you need so let's look at the first scan. You need anthropometry measurements, and that goes into a software. So you still need a radiologist to be able to do it. Only thing is that you will have a method which is based on your fetus, the Indian or the South Asian fetal anthropometry. If you use the images per se, you don't need a high-end radiologist. These images will then they'll be put into the software in such a way. We have an app which is very, very preliminary. Yes, yes, exactly.
So what I meant to say that they will give you the exact date of pregnancy.
It will give you a gestation period. Yes, yeah.
But to develop the machine learning, 1,000 sample will not be enough.
No, it's not. This is only validation. The machine learning was done on 10,000. Yeah. So this I'm saying is validation. 1,000 is the first validation. I said this is in CMC. We'll go to Northeast. We are already starting in Delhi. So this will have to be a huge validation. We are not going to roll out anything.
Different cohorts.
That's what I said. External validation. Yes. So this is the last of our AI program, and then I'll come to things which you're more familiar with, and maybe I won't be able to answer some of the questions there. But this is, we are using video sweeps of the ultrasound, which in the video sweep, which gets converted using, again, AI and machine learning into a risk assessment algorithm and into a traffic light decision system, which again will help us to triage in low resource settings. So what happens is that what we've got now is a breach detection with one video sweep converted into an algorithm. The breach detection at a small hospital is being possible. Again, this is initial data. We validated it in our cohort, but it needs to be, again, like you said, validated in the next step, and then, of course, put into a device for translation. So, coming to the biomarkers on omics platform, we have very, very dedicated partners who are working closely with us. I think our contribution to them has been because our clinical phenotyping has been very good. So we've been able to give them, we haven't given them samples, we worked on it together because we are phenotyping, and then this work is happening. So we have just finished the GWAS of spontaneous preterm birth from South Asia and India on 6,500 mothers. We've used study designs where we've actually done the selection in a very, very epidemiological way. So we've not just randomly selected. There's been a lot of thinking that has gone into this. And what we found is we've used a nested case control, and within the cohort, a nested case control from the 6,500 mothers, and used the global screening array for doing this work. And today we have we found 4 SNPs. There were 19 which were significant, and 4 SNPs which have showed a significant association both in Indian women and the European ancestral women. And this is the first time that this has been done in South Asia. It's under review in Lancet Regional Health. Hopefully we should have it published soon. And in addition, we've looked at a longitudinal study on spontaneous preterm birth in differentially methylated studies to look at what is behind spontaneous preterm birth. And we found some genes, but the 2
these listed here are those which have also been seen in preeclampsia and fetal growth restriction. So these are the two genes that we need to work on further, and what we are now doing is creating a RT-PCR based assay which can help identify these, to validate them, and also to be used as a clinical application if this validates well. So from our initial discovery research, using a smaller number of pregnant women and using the SWOT platform, we identified 60 proteins, which then have now been verified in a slightly larger number of preterm, and we have identified 8 proteins using the MRNMS analysis, and these proteins are now being validated in another 1,000 women to see whether they are actually going to be able to predict preterm birth. So this is one panel that we hope that after validation, and then of course, as you said, we will look at external validation, not only in India, but also in some of the international cohorts for this work. Similarly, we have worked on metabolomics, where we've identified using both discovery and again verification platforms, where we've identified, in the first discovery we identified 8 metabolites, and now using the targeted analysis we've identified 3 metabolites which are linked to spontaneous preterm birth, and they are going to, we've started the validation in a larger number of women now to take it further. So as I told you, we were doing vaginal swabs, and these were being done longitudinally. So we had the high vaginal swabs of these women, and what we found is, again, it's a busy slide. I think that's because my colleagues are very enthusiastic about wanting to publish, show everything, but just give you a little summary, that what we found is that Lactobacillus iners was significantly higher in all the trimesters in preterm birth samples, which is an important finding because our aims are two. One is to pick up a diagnostic biomarker, and the second is if we can develop a therapeutic, a probiotic, a therapeutic product which could help as the next step. And so this is what has been found in all trimesters. The other bacilli, the other microbiome, the other pathogens have been seen across term and preterm, but this is what has been seen across all trimesters of preterm. And we've had Bhaptosh and his group have created this dipstick to be able to pick up these different pathogens. And this again, we are going into a validation phase just now, because I keep telling them, just getting them in the lab is not enough. You need to go out. So we're talking now to the gynecologists, both in the district hospital and the other tertiary hospitals we work with, to validate this. And since Lactobacillus crispatus was found to have a lot of antimicrobial functions, this is being developed further. This work is happening on this to be able to develop it as a potential probiotic candidate. My last of this is, we have a we started a new program within this called the Human Placental Program. And this is because placenta comes at the end of the pregnancy. Everything has happened, and then you get the placenta. You don't know at what time point that placenta has actually been affected. And the placenta is the cornerstone of pregnancy. For all you know, many of the answers lie there. So what we are working on are surrogate markers, what we call the plaxomes, and the protocols, protein cargo, the protocols for protein cargo have been standardized, and this is Pallavi and her group, where top 20 candidates for deciphering the role of these genes in the placenta has been identified. And one particular protein, tenecin, is being seen in the ECM as in much higher proportion in the preterm placenta as compared to the term placenta. We are also looking at some initial work on whether the placenta harbors a microbiome. But this again is preliminary work and needs to move forward.
This one particular one, do you have the placenta weight you check on patients as well? Because they get
Yes, we do. Yes, sir, we do. So we are collecting placenta across all the women. We've tried to, as wherever they've delivered in the Gurgaon Civil Hospital. So we have huge numbers of placental punches. We can't collect placenta because that's too much. Our biorepository would not be able to take it. So we are collecting placental punches where it's all standardized. They've been kept in the regular storage protocol that we have. In addition, we are now doing the histopathology along with some research questions. Unfortunately, with the time, I cannot go too much into detail, but yes, we do have, you're saying for preeclampsia? Yeah, we do. Yes, but you'll be surprised, we found preeclampsia only in 6% of our women, because it's representing the population. Maybe at AIIMS you'll see much more. You'll see it much higher in the tertiary hospitals.
It's true that I may be totally wrong. You are a clinician. In Indian women, the preeclampsia comes in the second birth of the child, rather than in the first birth of the child.
That's what the evidence is showing.
Yeah.
And also, it is, but we don't know what the true reasons for that is. There's a lot of literature around it, but what we are seeing in the population is not really that much of preeclampsia, what you see in tertiary hospital. Having said that, it's a huge problem because it's reaching the tertiary hospitals. Yeah.
Dr. Singh, I have one question.
Yes. Yes.
It basically interacts with collagen and fibronectin, and essentially plays important role in bone remodeling. How do you correlate with your study why the expression of this gene is going up, and how you correlate it?
So this is a very initial preliminary finding that we found in all the 20 in the work that my colleagues have done, the 20 genes that they looked at, and they found this. Now this has to be, and they've seen it being expressed much more in the preterm placenta. So one will have to see whether there is some overlap in the kind of biology that you're mentioning. This is work that needs to move forward. And
so do you see the level of HCG, human choreogonadotropin, or TSH, FSH, or GPCRs, their level of expression is going up?
We are not seeing those hormones in all our patients, in all our mothers, but now specifically that we're picking up this story, it will be done prospectively.
Especially HCG or LH?
Yes, absolutely. I think it's important, and we'll do it prospectively. That's the idea of this whole long-term thing, that whatever questions are coming up here, we can take them forward. I'll just take a couple of minutes more, sir. Just to say that, again, an interesting story. When we started, nobody in the West was interested in this cohort because they thought that we are going to get the same kind of results as they've got. But now we are part of many global cohorts. The advantage, of course, is that we are able to harmonize our data, because what we get from 10,000 or 12,000 mothers will be actually, like now we're doing omics of the metabolomics in 26,000 mother-infant dyads across the world. So while our data is very important for us, it's also important for global knowledge that the numbers put together may give you a more definitive answer, keeping the heterogeneity in mind. So one will have to correct for that heterogeneity, but it's again very important that that multidimensional approach, that collaborative approach, is essential not only within the country but even globally. So, and my last slide, just to summarize. So where are we? We wanted tools. We've got, as I said, epi and clinical risk factor analysis. They've gone into policy papers. They are being published now. We have some very interesting ultrasound-based predictors and tools which are actually being validated and should hopefully go into clinical deployment next time I'm given an opportunity here to present. We have some initial dynamic prediction models, but the actual final result of these models will come only when we have all our 12,000 in, maybe more. We have, as I said, some omic-based biomarkers which are being validated. We have so we have a risk stratification algorithm which is largely here. Where the challenge is this area, and as you said, sir, we've got some data from the placenta. We need more biologists to come up and collaborate to look at mechanistic pathways, because we have very well phenotyped samples in the biorepository. We have well phenotyped pregnant women who we've enrolled, and we are going to prospectively enroll more. So this is an area which is a challenge, and we need much more collaboration on this. Having said that, some of my colleagues are doing a great work, and because of that, we've now been able to, we've got funding from the government as well as extramural to actually follow up the babies who are born to these mothers. Again, a very important way forward. So while we enroll the women, we also follow their babies for outcomes of growth, neurodevelopment, metabolic status, and mortality. And that, again, is going to be a very important national platform. So we have a Garbhani national platform for pregnant women. We have a Garbhani birth, a platform for birth and their follow-up, and of course the biorepository today. Just a last minute, not possible if all of us weren't together. And while Partha and Dinkar have been very, very strong friends and collaborators in this whole program, all my younger colleagues are here, the core group, and then of course these are teams. Today we have 200 people out there in the clinical teams doing this work. And that's not a small number, trying to keep them going, sustaining it, because you need to keep generating funds. And what we do is use this platform to get extramural funding. So today at this point, we have at least 10 grants which we've developed through extramural, both international and national grants, to do the smaller questions. And that's why we feel that it's very, very important for us to collaborate more and to reach out to audiences like this for more different disciplines to come in. And my special thanks to DBT, BIRAC, the Grand Challenges India, to WHO, to we have our global collaborators, above all to the hospitals and the study participants, and of course Professor M.K. Bhan, who will always be remembered for the very crucial scientific and technical feedback right in the beginning until when he was with us. Thank you.
Married very young in our country, and their bodies may not be able to support the babies. So do you see any difference between first birth and, let us say, third birth in premature or abortions?
Okay, no, first part, no, I'm sorry.
So the young girls probably may not be able to support
so you're
babies.
Saying abortions in younger girls?
Yes.
So our median age of mothers is 22. That's being enrolled.
But
so
that is not the married age.
No.
Usually girls they get married about 14, 15, 16.
No. So one of the things that we did was, because less than 18 is not legal, our ethics committee did not let us enroll any woman who was less than 18.
That is a technical problem, but scientifically this is what probably is more relevant.
So let me just answer that. So even if you're talking about, firstly we weren't looking at abortions, because that is something which has very different
sorry, I said abortion, I mean pretermination.
Okay. So even in the age group that we've seen, which is 18 and above, though I think they hide their age when they come and they tell us they're 18 and above, they would be much younger, but because they're not, it's a legal age that is, so, but
right.
we can't take less than 18. That's the ethics committee commitment from us.
I understand.
And when we take them, we do see a difference in age-related association with preterm birth. Yes, it is there. It's a known risk factor even earlier.
Are you typing the fetus, genotyping?
How do we?
Are you looking at the genotype of the fetus? Because that could be one of the important factors.
No, we're not. And that's a very important thing. And that's something that I can carry back. That's something which we've been asked earlier also, but we're not doing it.
Yeah, and to extend Subir's question, you're setting up your study in a clinic which probably caters to people of lower economic status. So are they really representative of what previous studies reported economically?
So we are not really low. We are low middle income because now it's good. Firstly, there were two challenges. One is that we want to represent the population as closely as we can when we start doing this. Ideally, it should have been the population. But we did a hospital, as I told you the reasons for doing that. It's not the poorest of the population. There is a low and a middle income group there as well. But I didn't understand why you would want us to, you're saying that this is representative only of a certain socioeconomic group.
No, what I mean is probably other studies maybe looked at different economic status in other countries. So can you draw anything because of this important difference?
So the upper economic strata, which will be say the Western population, they have a very high rate of cesarean section.
For a different reason, as you already shown.
So if we look at the higher socioeconomic, even in our hospitals, the rate of cesarean sections are much higher, leading to preterm birth and earlier gestation. So of course we can compare, but our idea was not to compare low and middle income countries. We were just following a time series event in the same group of mothers and different risk factors.
A couple of quick questions, yeah.
I mean, this is so much more difficult than what we do in the lab. Really admirable, Shinjini. The question I had is, where do you see interventions now? Let's say you come up with whatever tools you have to diagnose. Where do you see the intervention?
We are seeing interventions in our micronutrients, which I didn't present because that's far too, that is more clinical. And from the clinical side, we see interventions in micronutrients. We're seeing some interventions with our dating models here, because we're not going to be able to go into the clinic without showing a cause-effect relationship for picking up the early gestation. So that's the intervention. We see some interventions once we get the personalized models of pollution exposure, and at the maternal level, we see some interventions there. So those are the three interventions, and there are some more clinical interventions that we're looking at. We're seeing interventions with the microbiome and the infection status of the mothers. I didn't present all the data, but I'm happy to talk to you because there's way more data there which I couldn't put together. But that's a very important feedback for us also, because at the end we need to look at, crystallize those interventions in a more constructive manner.
Professor Shinjini, one more question. During your presentation, you talk about that biomarker, especially this kind of studies, therapeutically relevant biomarker detection will be that one of that appropriate target. What kind of approach you are thinking in order to identify therapeutically relevant biomarkers?
So I said one is microbiome, because that would be an important therapeutic biomarker for us. That's from the biomarker side of it. Even if we were to get the panels, the proteomic panels, which everybody tells me is a long shot. I don't know. It's something which whenever I've presented to a biology predominant audience, they say it's a long shot. But we've reached here, and we feel that when we need to look at it in the form of an intervention which is going to show we need a control group. So we will plan the next study of the evaluation of the biomarker in a randomized control study. We will not do it as an observational. So that's another intervention. We just need to show them in proper randomized control studies before we can take them for clinical deployment.
Last question.
Yep. Thank you, ma'am, for your presentation. I was wondering if there is any correlation between the number of pregnancies and the occurrence of PTP.
That's a known risk factor. Recurrent preterm birth and the larger number of pregnancies. All these are known factors. What I presented are some of the novel ones that we picked up, which are not being presented earlier.
Thank you.
Can we discuss during the tea time?
Yeah.
So I think.
He asked first.
Yeah. So please go ahead, Dr. Ramachandra. Please.
Why I am saying comment? Because this is giving us very good data. process. So there is SNP data. There is SNP data. There is high-end proteomics with SWOT analysis. There is vaginal microbiome data. And there is nutrition data. So now you need a very good, I think, artificial intelligence guy who should really make a very good program. Otherwise, it will become very difficult.
I agree.
SNP is for SNP or is it for vaginal microbiome or for the protein?
I wish there were more hours in a day. And I wish there were more people interested in this kind of
ask one question.
Finish it. With the data that you had on imaging microbiome and the biomarker, is it possible to get some sort of postmortem about the patterns right now which are correlated to early birth and then try to refine it as more and more data?
So you're saying combining integrated analysis, that's also what Subir is saying. Yes, that is the other main objective, which is one of the main things that we began with. But can I just take a minute? So, we began with that. We thought it would be as simple as, and I'm learning as a physician, that it's not that easy when you have data of different kinds and then you want to integrate it. To actually do that, it's even in the global cohorts they've not been able to do. That's I believe complicated stuff, but that's the next step forward. And we'd be very happy if people want to collaborate on that.
That would be a very important outcome about how.
Yes, absolutely. Yes, I agree. That's what we want to do.
So,
We're happy.
we will finish it, and I would like to thank all the audience and also Dr. Madhu and Dr. Shinjini. We worked together in INSA for so many years. So thank you very much, and please join us for tea at the Murthy Auditorium.
Thank you so much, sir. Thank you so much, Professor Jagannathan. And actually, we will now break for tea. And after tea, we are going to have... So we are going to have the Homi Jahangir Baba Medal Lecture at YVS Murthy Auditorium itself, and which will be followed by the session over here. We will be back here by 12:15 for the next session. Thank you so much. There is arrangements for